Okay, hi, and welcome to our first lecture on the central nervous system. So it's just going to be kind of an introductory thing. I hope that you are studying well for your exam that's due, and that that you do well on it. So let me go ahead and find. Oh, there we go. My PowerPoint, and there we go. Okay. All right, so in this, there we go. Okay, so in this unit that we are discussing, we're going to talk about the central nervous system and the peripheral nervous system, and all of their functions and where they come from and everything like that. So we're going to start out this lecture right now. We're going to watch the crash course video on brain development. Well, actually the whole central nervous system, but it has a good section on the process of brain development, and so I want to discuss that at some length. So we're going to go ahead and watch this as soon as it comes. There we go.
James was a healthy professional, a father of two. He had lots of friends, loved telling jokes, and played softball on Sundays. And one day, at the age of 45, he suffered a stroke. He bounced back fairly quickly, with one major exception. He was no longer able to speak. The stroke damaged a specific area in the left hemisphere of his brain called Broca's area and left him with what's known as Broca's aphasia. Broca's area is partly responsible for the ability to produce and process language, and Broca's aphasia often leaves its sufferers with some ability to understand speech, but an inability to produce intelligible words. James could understand his wife when she asked if he wanted cereal for breakfast, but he could only respond by repeating the word "to," although he could still intonate as though he were speaking the whole sentence. Then, after some time in therapy, something rather unexpected happened. James regained some ability to communicate through singing. Broca's aphasia can sometimes be treated by teaching patients to sing, because singing uses a different region of the brain, one that's on the right side and that's analogous to Broca's area on the left. So after some practice, James could sing words, and he eventually relearned how to talk by teaching the right side of his brain to speak rather than sing. Whether it's a stroke affecting your speech, a tumor destroying your memory, a concussion affecting your aggression, or that fateful iron rod that shot straight through Phineas Gage's skull, a lot of what we know about how the brain works has come through studying injuries to it. And what we've learned so far is that even though it looks like a 1.4 kilogram lump of gray congealed oatmeal, the brain is made up of super specific areas that have super specific functions. You might actually say the same thing about your brain that's sometimes said about politics. Everything is local. Nervous system is You'll remember that our nervous system is divided into two main networks that work in harmony: the central nervous system, consisting of your amazing brain and spinal cord, and the peripheral nervous system, made up of the nerves coming out of that central nervous system. The central nervous system's main game is integrating the sensory information that the peripheral system collects from all over the body and responding to it by coordinating both conscious and unconscious activity. The sun is bright, so I'll shade my eyes. I'm hungry, so I'm calling the pizza man. The phone is ringing, maybe I'll answer it. All these sensations, thoughts, and directions processed through this two-part system. It's the brain, of course, that sorts out all that sensory information and gives orders. It also carries out your most complex functions, like thinking and feeling and remembering. Meanwhile, your spinal cord conducts two-way signals between your brain and the rest of your body, while also governing basic muscle reflexes and patterns that don't need your brain's blessing to work. This is how a chicken can still run around even if the poor thing has been decapitated. Both your spinal cord and brain are made of fragile, jelly-like nervous tissue that is extremely susceptible to injury. So all that goo is well protected by the bones of your vertebrae and cranium, as well as membrane layers or meninges, before being bathed in a cushy waterbed of clear cerebrospinal fluid. This fluid actually allows your brain to float somewhat in your skull, reducing its weight and letting it slosh around while you and your head are free to move. But even with all that extra protection, your brain is still vulnerable, and one thing that James's story taught us is that its vulnerabilities can be incredibly specific, because your brain is divided into specialized regions regions that may or may not interact with each other to produce a given action. We can better understand this division of labor by looking at how the brain first develops into its main component parts. Inside a developing embryo, the central nervous system starts off as a humble little neural tube. Soon, the caudal or lower end of the tube stretches out, forming the spinal cord, while the cranial end begins to expand, divide, and enlarge into three primary brain vesicles, or interconnected chambers. This is kind of your proto-brain. We call these chambers the prosencephalon, the mesencephalon, and the rhombencephalon, or forebrain, midbrain, and hindbrain. By an embryo's 5th week of development, these main three chambers start morphing into 5 secondary vesicles that essentially form the roots of what will become your grown-up brain structures. The prosencephalon divides into 2 sections, the telencephalon and the diencephalon. The rhombencephalon forms into another pair called the metencephalon and the myelencephalon. And in between, the mesencephalon, thanks to evolution, remains undivided. The real action starts as these 5 secondary vesicles start developing into the major adult brain regions that you might be more familiar with: the brainstem, the cerebellum, the diencephalon, also known as the interbrain, and finally the cerebral hemispheres. But in order to go from the simple tube into that classic wrinkly icon we think of as the brain, each of these 5 vesicles grows in different ways. Basically, some develop a lot more than others. The least dramatic changes occur in the 3 most caudal or lower sections: the mesencephalon, the metencephalon, and the myelencephalon. They go on to form the cerebellum, which mostly helps coordinate muscular activity, and the brainstem, which plays a vital role in relaying information between the body and the higher regions of the brain. The brainstem actually has 3 main components, and I know this is going to be a lot of vocabulary here. You have the midbrain, the pons, and the medulla oblongata. Together, they regulate many of the most basic vital involuntary functions, like keeping your heart on pace, keeping your lungs working, and controlling things like sleep and appetite and pain sensitivity and awareness. But of the 3 brainstem parts, it's your midbrain that carries out the higher level functions. Like when your eyes track a fast moving object, or when you look behind you after hearing some sudden loud sound, it's the midbrain that receives and processes that sensory information and sends out the reflexive motor signals so you react without thinking. The midbrain also passes that data to regions like the cerebral cortex, which do the actual conscious thinking about the stimuli, like, what is that thing whizzing across the sky? Or what just exploded behind me? So with the brainstem and the cerebellum covering your basic life and motor functions, you start to see somewhat more complex tasks being carried out in the next major brain structure, the diencephalon. This is where you find the thalamus, hypothalamus, epithalamus, and the mammillary bodies, which regulate things like homeostasis, alertness, and reproductive activity. Here we also find part of the limbic system, which is a center for strong emotions, like fear. This area is sometimes called the reptilian brain because we share it with some of our less philosophical animal brethren like lizards and fish. I'm not putting these guys down, but by our standards, they don't think so much as focus on the more instinctual pursuits that are ruled by the caudal regions of the brain: eat, drink, sleep, mate, stay safe. All those things are awesome, but it wasn't until the appearance of birds and mammals that some animals' brains came to be dominated by the last of the 5 vesicles, the telencephalon. During your brain's growth, the telencephalon undergoes the biggest changes of all as it develops into the most brainy part of your brain, the 2 classic walnut-looking hemispheres we collectively call the cerebrum that cover the rest of your brain like a mushroom cap on its stalk. That's the cerebrum, not to be confused with Cerebro, which is Professor X's telepathy-enhancing device, and it is the largest region of the brain and performs the highest functions. It's made up of the wrinkled outer layer of gray matter called the cerebral cortex and the inner squishy layer of white matter beneath it. And it's the cerebrum that rules our voluntary movements and our most advanced tricks, like thinking and learning and regulating and recognizing emotions and experiencing consciousness in general. You'll remember that higher processing requires lots of synapses, which require lots of nervous tissue, so as the cerebrum grew through evolutionary time, it got more massive, but our skull didn't exactly keep up. So in order to squeeze all that material into your skull, the brain forms little creases called gyri and larger grooves, or sulci, giving it more folds than an origami pineapple. And although a big fissure separates the left and right hemispheres, the 2 halves communicate through a series of myelinated axon fibers called the corpus callosum. And each hemisphere has other smaller fissures that divide it into lobes, each with a different set of major functions. The frontal lobe, for example, governs muscle control and cognitive functions like planning for the future, concentration, and preventing socially unacceptable behaviors. In most people, this area doesn't finish developing until after the teenage years, which tells you a lot about the teenage years. Since Broca's area lives in this lobe in the left hemisphere, it also is important important in language comprehension and speech. If you're enjoying a beautiful sunset, you can thank your occipital lobe at the back of your head for processing those bright visual cues. And the next time you step on a Lego, you can curse your parietal lobe, which processes the sensations of touch, pain, and pressure. Meanwhile, the temporal lobe helps sort out auditory information, including language. It contains Wernicke's area, another important region of the brain associated with the production of written and spoken language. This part of the limbic system includes your short-term memory keeper, the hippocampus, and the emotional amygdala, which controls sexual and social behavior. So if you damage the wrong part of your temporal lobe, you may never again be able to remember what you ate for lunch, or you might suddenly become a total jerk who kicks kittens and cuts in line. We could do a whole course on the finer-grained functions and consequences of malfunction in every bit of brain in your gourd, but, well, we can't do that today. And you gotta remember that when it comes to your body, no organ or system is an island. Your brain would be pretty useless if it weren't hooked up to the outside world. That's where the peripheral nervous system comes in, which we'll be spending the next few lessons exploring. Meanwhile, you learn.
Okay, so I suggest that you take your notes sometime that we'll be looking at here in the future, and the rest of the notes that you have for the central nervous system, and go back and watch the video and then match up the structures with their functions, because yeah, like he said, there's a lot of vocabulary, there's a lot of terms in this section, because there's not really how things do this. It's mostly just what does what's responsible for what in your brain. And so it's a really good idea to rewatch that. I'm also going to make a page that I'll put in with the in the module that will give you different resources that you can go to, different pages and things like that, that you can go, websites that will help you with this. Okay, so what we need to do right now is take a look at how did that tube form originally when that became your brain. Okay, so once upon a time when you were an embryo, and I'm going to try to find a video, I think, while we're still in class today, of how this new neural tube formation works, and then what happens when it doesn't. I was trying to decide if I wanted to show it today, but I think I will. So, or maybe I won't. Maybe I won't. Maybe I'll save that for Monday. Yeah, I think I'll go ahead and save that for Monday. So let's just talk about what's in your notes right now. Sorry, sometimes I have to plan out loud. Okay, in the middle, in the moment. All right, so let me draw first of all what you looked like when you were an embryo. All right, so you looked like, I'm going to do two, well, actually, let me clear that. Let me do it 3 different ways. Okay, so we want a side view of you as an embryo. I know you're not very appealing looking. Okay, and then a top view. And then a cross-section view. Okay. Yeah, this is you were so cute back when you were 8 weeks old. All right, so we used to be made out of 3 different tissues, just 3 basic tissues. So I'm just going to color sections right here. So right here along the oh, let me undo that. Undo. Okay, let me do it this way. Okay, this is better. Yeah, this is much better. All right, so you had 3 layers of tissues in you. So this was your first layer. This was called ectoderm. And then you had a second layer called mesoderm. Filled all in here. Whoops. And then all in here too. And then you had a third layer called endoderm. And endoderm would have surrounded this tube right here. Okay, and so all of this would have been filled in. There wouldn't have been any white. There would have just been all of these colors. Okay, but the one that we want to take a look at is this one that's on the outside of your back called ectoderm. So this is all, this, because we're looking at you from the top, all of you is ectoderm here. Okay, so all along your back was all ectoderm. And then this is ectoderm right here. And this is maybe the one that we want to use the best, is this picture. So this is your abdominal cavity. And so this is your abdominal cavity. This is your gut. Okay, and then everything else in here, all your bones, all your muscles, all your heart, all your circulatory system, everything like that will come from here. But this part of your back right here began to fold in. And so we had a dip. That's kind of a deep dip, but we're going to, oh, I didn't want to erase all of that. Okay, well, I'll just put it back. Okay, so this here was your outside of you. All right, so it started to dip down like this. And as it dipped down like this, let me show you another picture. It came together and then it broke off. And so here was your cross section of you, and then here is your spinal cord. And then back here, this was your gut tube. Oops, it descends like that. And then your abdominal cavity. Okay, and then you'll sprout arms and legs off of here. And then you would have a head up at this end, and then your tail. Oh, you look like a turtle. All right. So this is where your nervous system started from. And so what your nervous system would have looked like, if we do this, then you would have had down your back this tube that would have started here, and it would have run all the way down here and all the way back up here. And so off of this tube, we're going to turn this end, because this is your head end, this is your tail end. Okay, up here, this is going to turn into your brain. And then coming off of here is going to be all your spinal nerves that we learned about. All like that. Okay, so let's get rid of my horrible drawing things now. And let's go back to this. All right, so how are we going to go about, so that blue stuff that I drew that was on the outside is called ectoderm. So it's going to form a neural plate, it's going to fold to form neural folds, and then a groove, and then a tube. So here is a maybe much better picture or depiction of it, but anyway. So here is in your back. So what we did was make a groove right down here in the middle of your back. Let me make that. Okay, so here is the groove. So we just made a dip right there. That's this dip right here. Okay, so about, oh, did I say 8 weeks? No, you're done by 8 weeks. At 3 weeks, well, even before 3 weeks. So 12-ish days, something like that. We're going to start by 19 days after you've been conceived. Okay, so your mom is barely late. If your mom had a regular period, she would have got pregnant on day 14 of her cycle and then started her period on day 1, which would have been day 29, so 15 days after that. And so she just is maybe barely considering that she might be pregnant, and your nervous system is already starting to form. Okay, so then that dip, this neural plate, is going to deepen to become the neural groove, and then we're going to have these folds that are going to come in here, and plus this folding of the ectoderm as well. And then it's going to come around. Okay, and then eventually we'll have what's going to form your skin over the outside of your back, and then your neural tube. Okay, and then these somites will end up becoming like your arms and your legs and bones and other kinds of things too. Well, no, those are neural crest cells. That's something else. They become other things we'll get into later. Okay, so here is this neural tube. So now the neural tube is going to get, subdivided. So once it's just a plain old tube like this is what we started out with. And then it's going to get into 3 swellings, or we call vesicles. And they're the prosencephalon, mesencephalon, rhombencephalon, like he said in the video. So they're just forebrain, midbrain, hindbrain. Don't really need to remember these names. Just know that we're going to start to get some enlargements here of this tube. And notice, because it's a tube, it's not hollow. I mean, it's hollow, but it's going to be filled with cerebrospinal fluid. So all of this blue here is all cerebrospinal fluid. Okay. And now we're going to get modifications to these 3 sections. So if we look at this section right here, the prosencephalon, it's going to really be modified to become the telencephalon and the diencephalon. So this is the part that we think with. This is our cerebrum, our cerebral hemispheres. The diencephalon, like he mentioned, is all of our autonomic control. So everything that takes place without us thinking about it will happen through the diencephalon. So these are 2 very, very important regions of the brain. Then, like he said, the mesencephalon doesn't really change at all, the reptilian brain, so this is just the middle part of our brain. And then the rhombencephalon specializes, you know, to fit our needs as human beings. And so we get the metencephalon becomes the pons and the cerebellum, and then the myelencephalon is lastly the medulla oblongata, and then the rest of the cord, so from here on down, is pretty uniform, like we saw in lab this week as we went through and did the spinal cord. So then the rest of the neural tube will then be the spinal cord. All right, now one other thing right here, it says adult neural canal region. So this hollowness inside of here, these are, you can see they get big. I get these big, like Mickey Mouse ear kinds of things. Those are going to become the lateral ventricles. Then I have an opening, a tube in the middle of my diencephalon that's bigger, called my 3rd ventricle. Then it's going to be connected to my 4th ventricle right here by this little tube called the cerebral aqueduct. And then this, not big, you know, ears at all, like in these other 2 regions, but just these little dips out here are going to be called the 4th ventricle. And then running down the middle of the spinal cord, like we saw in lab this week, is the central canal. And then the central canal plus all of these would all be lined with lined with ependymal cells and have cerebrospinal fluid in them to keep circulating the cerebrospinal fluid. Okay, so yeah, on Monday, I'm going to show you a couple of videos more about this information instead of going and looking for it today. I'll do it on Monday. Okay. So now here is that same kind of thing that he showed you in the video of what happens, you know, modifications that happen to your brain. So we can see the spinal cord is the one that really stays just pretty much like that original neural tube. Then we start to get folds and bends as the tissue grows. And so at 5 weeks, we have, you know, not a lot has happened to the myelencephalon, let's see, myelencephalon. But now we're starting to get a little out-pocketing here in the metencephalon. And then we have mesencephalon is the part that really doesn't change very much at all. But now this one, we're getting some growth. Anytime that you grow more, it's going to make this tube fold up on itself. Like he said, your skull doesn't give much room for this neural tube to grow and grow and grow and grow inside of. And so as it grows, then it just folds up on itself. And then eventually we get the most growing and folding in the telencephalon that will then become the cerebral hemisphere or the cerebrum. Now, the reason why we want our neural tube to grow, why we want the telencephalon to end up looking like the cerebral hemisphere with all these folds and ridges in it, is because we want to increase the number of neurons that we can have. And you can only put them, they only come on the edges of the tube. So what we want to do is lengthen the tube and then just coil it up on itself so that you can fit it inside of your brain or inside of your skull. And so that's why our brain is so convoluted, is that just we had all of this growth, this expansive tissue growth happening on the very caudal end, the rostral end or the cephalic end of the neural tube. And we just scrunched it all up so that it would fit inside of your skull. All right, so here are the 5 sections of the parts of the brain. So I don't care so much that you remember telencephalon. Maybe remember diencephalon, you'll hear that again, meson. So when I learned these, when I was in college, I learned from the neural tube, so from the spinal cord up. So I learned my met, mes, di, tel. So whenever I'm thinking about it, I have to go my met, mes, di, tel, and then I can remember the different sections of the neural tube. But what I do want you to know, though, is what things go together. So if we take a look at, let's do this. Okay, so if we look at telencephalon, cerebrum, cerebral hemispheres, and lateral ventricles, okay, those are all a unit. So those all go together. Okay, then the diencephalon, thalamus, hypothalamus, epithalamus, third ventricle, those all go together. Okay, then midbrain, corpora quadrigemina, and cerebral aqueduct, those go together. Okay, and then brainstem, pons and cerebellum, and fourth ventricle, those are a unit. And then the medulla oblongata is its own thing. Okay, so just remember what formed together and what things are associated with one another. All right. If you want to read more of a description of the major brain regions, you can go ahead and read through these, but I'm not going to worry about that. Okay, so let's take a look at the ventricles. So the ventricles are, let's go back here. These are the out-pocketings, okay, the little pouches that stick out from this original neural tube. Okay, so here's the original tube, and we modified it here, and then we made the little Mickey Mouse ears and whatever. Okay, so these parts right here, so these two things right here, and the tube that connects them, and then these things right here, and the tube that connects them, that right there, those are all the ventricles. But look at what they look like now. They don't look much like they looked in that other diagram, because they've been so mature to the point where here we have an adult brain, and this is what they're going to look like. Now remember, a ventricle is always something that's going to hold fluid. So if you think of the ventricles in your heart, okay, the left and right ventricles have blood in them. So the ventricles in your brain have cerebral spinal fluid. And we'll probably get into them more in lab on Tuesday, but let's just remember what the names of them are. So these two right here, you have 2 lateral ventricles. You have 1 in each cerebrum. Okay, so they bear their own ventricles. Each cerebrum bears its own ventricle, cerebral hemisphere. And they kind of look like ram's horns, like bighorn sheep horns. Okay, and then they're going to be connected together to one chamber right here in the middle, kind of looks like a little face thing, called the third ventricle. So we have the first and second are the 2 lateral ventricles. Then we have this little interventricular foramen, so a little hole that's going to connect the cerebrospinal fluid in these 2 ventricles with the third ventricle. Then we're going to go down through this tube right here. Okay, so this tube right here is called the cerebral aqueduct, until we get to this fourth ventricle, excuse me, fourth ventricle that's at the level of the cerebellum right here. Okay, so those are the ventricles of the brain. And again, we'll probably talk more about them in lab on Tuesday. So they're the openings of the brain. They are continuous with one another because they all can make cerebral spinal fluid. So the 2 lateral ventricles, the third ventricle and the fourth ventricle, contain structures called choroid plexuses, which are capillary beds that basically leak out plasma to become the cerebrospinal fluid. And then remember that they will also contain ependymal cells. They'll be lined with ependymal cells so that they will create a current that will always flow, which we'll be looking at in the future. And then we have the 2 lateral ventricles, third ventricle, cerebral aqueduct, and fourth and central canal. Ooh, that should have a comma right there. So lateral, which are first and second, third, comma, cerebral aqueduct, and fourth and central canal. Okay, so those are the ventricles. Now also, we'll talk about that on Monday, so we'll come back to that. All right, so if we take a look at this side view of the brain, we can definitely see the two major sections to the brain. We have the cerebrum, so that's this big part all up here that's all convoluted. One thing that I want you to pay attention, if you remember when we were doing the 3 parts of, sensory input, integration, motor output. Remember we said sensory is always going to be blue and motor is always going to be red. So sensory input's always blue, motor output's always red. So you can see here these 3 parts of the brain. So the parietal lobe, occipital lobe, and temporal lobe, see they're all different shades of blue. That's because they're all involved in sensory input. So information that we collect from the outside world is going to be brought into these 3 different parts, or 3 different lobes of the cerebrum. So temporal lobe, occipital lobe, parietal lobe, just like the bones under which they lay. Okay, so you've got temporal bone, occipital bone, what did I say first? Temporal bone, parietal bone, occipital bone, and then underneath them will be the corresponding lobes. So this is all sensory input of different kinds. We got different kinds of sensory input, but that's all going to be coming into those 3 regions right here. But this is all red, it's pink, but this is red. So this must be motor output. And then inside of here will be the integration, where the interneurons are. We'll learn about that next week. Okay, so this is all motor output. So all of the voluntary movement that we do is all going to come from somewhere in this red frontal lobe. So all of this thing is the frontal lobe. All right, now I'm going to go ahead in our notes and then I'm going to come back to that picture. All right, so we notice when we take a look at our cerebral hemisphere, so there's a real brain, and then here's cerebellum down here. When we take a look at the whole brain, obviously we see all these ridges. So they're all the lighter colored stuff, the tan colored stuff. And then we have these grooves, and there are blood vessels that run all in the, cerebral hematoma. I knew there was some other video that I got to put in with the ones that we're going to watch, and it just popped into my head. Okay. So blood vessels that all run here within the grooves. Okay, so we've got ridges and grooves that come as a result of that neural tube growing, and being all shoved up inside of your skull. So what are the ridges and grooves? The ridges are called gyri. So, and we'll take a look at what this means in a minute, where it says gray matter or cortex on the surface and white matter underneath. Increases the surface area for neurons, like I already said. Now we have what's known as a precentral gyrus, and this is where all of our voluntary motor recruitment originates in the frontal lobe. I'll explain that in a minute. And then we have a postcentral gyrus for somatosensory perception in the parietal lobe. Okay, so I'm going to explain all of that when we go back to the picture we were looking at in a minute. And then sulci, so we have gyri and sulci. Sulci are the grooves in the cerebrum, also for the surface area. So when you think about it, if you took a garden hose and you folded it all up and shoved it inside of a skull, you would have ridges and grooves. So the parts where when you bent the hose to put it together, to wrap it together, or you rolled it up in a circle, the places where the hose touches a different loop of hose, that would be a sulcus, and then the hose itself would be the gyrus. All right, so what we did in this picture is we cut a chunk out through of the cerebrum, and here's what we see. So here are the ridges right here, and then so the folds, and then the grooves where the folds touch each other are the sulci. So a gyrus and a sulcus. Now notice that there is a darker colored tan material right here we call gray matter. So it really looks brown in a human. So I don't know why they called it gray matter, but they did. And this is gray matter. We'll talk about what gray matter is in just a minute. And then we have white matter. So I want to show you a couple more pictures of gray matter and white matter. Find them. Okay, so okay, this is good. This is a good picture. So what we did, this is looking down on top of your brain. So here is the front, here's the back, this is the left side, this is the right side. Notice how the outside part of our brain is all this gray matter, and it goes around every gyrus, and it goes over every sulcus. So here I've got sulci, but it goes around every sulcus, goes around underneath every gyrus. Okay, so there's where all the gray matter is, and you can even see in this actual brain picture. Here's all the gray matter out on the edge, and then this white stuff in here, obviously, is white matter. So here is the white matter. And then we have some extra gray matter in that we'll talk about later on, okay, what these extra gray matters are. All right, so gray matter is unmyelinated axons, dendrites, cell bodies, and interneurons. So everything that's unmyelinated is going to be up here in the gray matter. And everything that's myelinated, so axons, are all going to be white